pictures help you identify where it is in the anterior neck, just inferior to the thyroid cartilage. Thyroid gland in the anterior neck. Inferior to thyroid cartilage. The thyroid cartilage is your voice box. I mean, in anatomy, we learn it as the larynx, as the major cartilage of the larynx. There are other ones. So basically, the word thyroid means shield shape. So the thyroid cartilage has the appropriate name. It's basically like a big shield, this whole structure there. So again, that prominence, the laryngeal prominence, is the Adam's apple in males, and the gland is what we're talking about here. There it is, kind of like more in the neck with everything else in a dissection. So that's the location of the gland. And here's a picture where you can kind of see it from the anterior view, just below the thyroid cartilage. That's the hyoid bone. This is a posterior view. These are the constrictor muscles of the pharynx of your throat, and that's the esophagus there. But this is the gland from the posterior view, and these four nodules, these two paired nodules here, and these two paired nodules here, I guess you have an inferior pair and a superior pair. Those four are the parathyroid glands. Two superior, inferior, parathyroid glands, located posteriorly on the thyroid. They're like within it. I mean, they're different glands, but they are located within it. Now, we looked at this under the microscope the last time. So, quick, easy question. I'll put an X there. The slide seems a little different from the one I showed you yesterday, but what do you think that is? Thyroid or parathyroid, where the X is? It's thyroid. Okay, the big purple splotch with all the chief cells, that's the parathyroid. There's no follicular arrangement here, right? It's the follicle arrangement that's associated with the thyroid, which I remember my lab instructor for anatomy, he called them like little steaks, depending on how you stain it. I mean, just that bright flesh-colored pink surrounded by the follicle cells, it looked like little steaks, and that's the general structure of thyroid follicle, which we've talked about already. And if you have to identify cells outside of the follicle, okay, those are the parafollicular cells, and they stain a little different here. So this one actually kind of looks more similar to our slide, I think. Okay, so let's kind of review the hormones that are secreted there.
I have a quick question.
Yeah.
On the quiz, can you write C cells or chief cells, or are you that not acceptable?
Yeah, you can write C cell.
Okay.
But the chief cell is not a C cell.
Right, I know.
Yeah.
But I'm just asking if we could use those.
You can use C cell for parafollicular cell.
Okay.
The C stands for clear, because histologists thought the cytoplasm of those cells was more clear than the follicle cells. So C cell is acceptable. Okay, so in the thyroid, it's the follicle cell that secretes thyroid hormone. Okay, and it's the parafollicular cell or the C cell that secretes the calcitonin. So those are two different hormones from this gland that we study, which are responsible for. Here's a simplified picture of the follicle with the parafollicular cells outside the follicle. Okay, just so you can easily see T3, T4. There's another way to refer to thyroid hormone, and we'll talk about the function of calcitonin. So here's the parafollicular cells, and when they secrete calcitonin, their mechanism is to lower blood calcium. Okay, so that's the function of this hormone here. So the physiology, the mechanism of how it does that is shown in those bullet points. Number one, calcitonin inhibits osteoclast activity. Calcitonin, that's one. Inhibits osteoclasts. If you remember, those are the cells they're multinucleated, and the ruffle border secretes all these lytic substances that dissolve the inorganic matrix of bone, liberating calcium, which would increase blood calcium. So if you inhibit these cells, that should lower blood calcium, okay, because they're operating less. That's the thinking there. All right, so you promote calcium uptake into the bone matrix. So you're depositing calcium into bone. That'll lower it. Increase calcium deposition. Into bone. And thirdly, you increase the excretion of calcium at the kidney. Increase calcium excretion in the kidney. So, again, we have a kidney, so make sure you understand that. Kidney. A little indentation part is called the hilum. Where you have blood being filtered. So the renal artery is applying all the blood that the kidneys can filter. So blood goes in, along with the calcium, that whatever amount of calcium you have in the blood. So basically, blood goes in, blood goes out. Arteries supply the blood. The veins return the blood to circulation. We talked about last time. The blood comes out. Question becomes, what happens to the calcium? If the body conserves it, it'll take it out of the filtrate and it'll put it back into the blood, and it will not be excreted. So calcium, make it back out and stay in the body's circulation. However, if it's excreted, it'll go out the ureter. Into the toilet, urination. Okay, so the question becomes, well, what am I talking about this for? Because this. So the key physiology word is excretion. That means a very specific thing in physiology. The kidneys are the only organ that have this function. Okay? If something is circulating in your bloodstream, the kidney is the only organ that can filter it out and excrete it. And you lose it to the urine. So, that's why. That would lower blood calcium too. Okay. All right, so the other hormone. Yeah, you got a hormone that can, lower blood calcium. You need a hormone that can raise it. Calcium function is very important for muscle function, for example. So this slide is trying to teach students the mechanism of how PTH does the opposite of calcitosis. I'm going to erase this now. Now, this is a good figure. Now, if you look at it, they're trying to show you by symbols that calcium is the little red balls. And the PTH secreted by parathyroid hormone, the parathyroid hormone is PTH secreted by a parathyroid gland. Those are the blue triangles. Parathyroid hormone secreted by parathyroid gland. Stimulated by low calcium levels. If calcium is low, that will stimulate PTH secretion. And that's shown. If you look on the right-hand side, they illustrate very few red balls. So that's a direct stimulus to release PTH in the bloodstream, and they showed parathyroid hormone acting on all of its targets here, here, and here. So when it acts on bone, it says it activates osteoclasts, and so you release more calcium into the blood. So number one, activate osteoclasts. I'll just kind of draw another picture of it there. So if you increase their activity, that should increase blood levels of calcium. Secondly, PTH increases calcium reabsorption. So you decrease its excretion. Increase calcium reabsorption at the kidney. So when you reabsorb it, you don't lose it to the urine, you keep it into the bloodstream. Okay? That'll also increase blood calcium levels. And the last one, at the gut. It passes through your gut. If you don't absorb it, you lose it to the feces. So through vitamin D, PTH promotes vitamin D, and vitamin D is the molecule that actually can increase calcium absorption from the gut. Parathyroid hormone via vitamin D some books call the active form of vitamin D calcitriol. So if you read that word in any of your studies, same thing, vitamin D. So anyways, through vitamin D calcitriol, you increase calcium absorption from the gut. All three of those increase blood calcium level. So if you look at the figure, after these 3 places in the body, and you put more red balls into the blood, so they show more red balls on this side, right? And that rising levels of calcium should inhibit its release. So that's kind of how you study that figure. So this is kind of a mental gymnastics. On your half sheet, let's start to remember how you handle a multiple choice question. Okay. Have you ever thought about that? What is a multiple choice question? I put that as a prompt. So what do you do with each choice? It's basically like a beefed up true-false. You have to evaluate each choice if it's true or false. And you love this, right? Oh, it could be both of them. I gotta consider both carefully. It could be just one of them. On your half sheet, call this number 1. Answer this. Kind of a mental gymnastics. I'm just curious, what did you guys put for that? Yeah, for different things. So here's how, how you do it. See, that actually makes sense. Okay, first of all, you have to remember, what does that do? What does PTH do? Does it raise or lower calcium? Raises. If you can't remember that, you can't answer the question. But if you did remember that, you have a chance to get it right. Well, I guess you could guess and get it right if you didn't know. It raises it. Okay, does this make sense? That makes sense. If those cells increase their activity, you'll get more calcium in the blood. This doesn't make sense. Because if you increase their activity, that doesn't jive. That doesn't make sense. So you X that out. Okay, if you increase calcium excretion, will that raise calcium? Well, no. You're going to lose more to the urine. So this doesn't make sense either. Which makes that no good. Okay, so that's the kind of thing you have to do. These kinds of questions. And do this one too. Number 2 on your half sheet. And this time I'm not going to walk through it with you. Just see if you can get it.
means deposits.
Yeah.
All right, I want to move on.
I know, it's like,
Let's make sure we hammer out all the details of thyroid hormone. And then I'll talk about how it's synthesized by the follicle cell. So thyroid hormone, TH, comes in two forms, T3, T4, so know both name and T4. So in other words, thyroxine is also known as T4. Triiodothyronine is T3. Turns out that's pretty much the active one, okay? The 3 and 4 refer to the iodine molecule stuck to the tyrosine residues, so let's go. You get another comment. Tyrosine is an amino acid and it's part of the thyroglobulin. Tyrosine is the amino acids on thyroglobulin that are iodinated. So let's draw tyrosine as just a little, pink bar. So think of two tyrosines together, pink bar, and if you draw some iodides on it, that would be a T4. If you knock one off when you get close to the target tissue, that becomes the active form. So there's an enzyme that could do it. So T3 would be illustrated as just 3 of them on it. So T4 is the one that's mostly secreted, but when it gets close to the target tissue, most of T3 is converted from T4 within the blood circulation. So here's a picture of, here's a slide showing you the effects of T3. The main thing that I mentioned before, it boosts metabolism. And when you do that, you have an ability to lower blood glucose. That's important. Let me note that. Increase the basal metabolic rate. That has the effect of lowering blood glucose. There's not too many hormones that can lower blood glucose. This one and insulin, that's it. Okay, a lipid-soluble hormone. So, it has receptors everywhere. The cytoplasm, nucleus, mitochondria. If you're going to boost metabolism in the cell, you'll turn it on some more. It makes sense that receptors are all over the place in the cell. Like we've been saying, you mostly secrete T4, 90%. Okay, most of it will be converted to T3 when you get close to the target tissues, it's varied. There's many tissues, mostly skeletal, cardiac, muscle, okay? And the stimulation, well, it's under TSH control, okay? So it's anterior pituitary gland dependent. TH formation is under TSH control. Falling T4 levels will stimulate TSH release. Rising T4 levels will inhibit its release. We think we discussed that before. So, it does require iodine. If you lived in, if there are landlocked populations that don't have fresh water, not seawater, and so the salinity of their diet is decreased. I don't know if you can see a little goiter there. It's because if you have low thyroid hormone levels, the pituitary through TSH will keep stimulating the follicles to make thyroglobulin, but with no iodide to extract from the blood, the follicles just get bigger. And so they call that a colloidal goiter. Okay. Well, here's a picture of how the cells can do that. Extract iodide from the blood. There's a follicle cell doing its job. And at the border between follicle colloid on this side, you can generate T3, T4. So let's kind of go through the steps here. So what you see here, they don't have it in the figure, but it's all being stimulated by TSH, thyroid stimulating hormone. So this is thyroid hormone TH synthesis under TSH control. Step 1. So once again, follicle cell, colloid, blood vessel. Where is there step? Step 1. Okay, under TSH control, you're going to make thyroglobulin. So this is the, ER Golgi complex that synthesizes proteins. Make thyroglobulin. And then you just kind of like, they show it excreting the tyrosines. They don't show the whole thyroglobulin. They just show the tyrosines, these little bars there, okay? Secrete to colloid. So if you want to identify colloid, you could call it colloid or you could call it thyroglobulin. That's basically what it is. So, those little thyroglobulins, I drew, it was kind of drawn like this, just the tyrosines. So that's how they illustrate it, just those little purple bars in step 1 right there. Now in parallel, you have to then trap iodide right here. Number 2, iodide is trapped and transported into the cell from the blood. Number 2, trap iodide from blood. You should bring it into the follicle cell and they illustrate it as green balls. So you trap it. Step 3 is then you have to oxidize it into the correct molecular form. Iodide is oxidized over here to iodide. So they change the color of the ball from green to blue to illustrate that it's been oxidized. Oxidize iodide to iodine from green. Iodide is, I think the correct molecular formula is negative charge. Iodine is I2. Wait. That is what you can kind of attach to the tyrosines there. So that's step 3. That's what you do in step 4. In step 4, iodine attached to what they call DIT-MIT. Step 4, I'll draw it over here. Step 4, you iodinate the tyrosine amino acids. So I'll just write iodinate tyrosines. So MIT, monoiodotyrosine, I just call it MIT. It's simply a tyrosine with one iodide molecule. So then DIT is the same thing with two. Diiodothyronine. That's step 4. Step 5, conjugate them to make T3, T4. Or moving on to step 5. Conjugate. MIT and DIT. So if you go MIT plus DIT, that'll give you a T3. There you go, so as simple as that. Then for T4, obviously you could figure it out, right? DIT plus DIT. DIT plus DIT gives you a T4. All right, so that is conjugation. Now you have to bring that back into the follicle cell to secrete it. So moving on from step 5 to step 6, thyroglobulin colloid is endocytosed, and you combine it with a lysosome to clean it up. So notice how they illustrate the colloid as this yellow goo. And you kind of have to free it from that. So what they do is they take the lysosome, which has lytic enzymes, and it leaves the T3, T4 intact, and they get rid of the yellow goo from there to there. So moving on to step 6. Endocytosis, T3, T4, fuse with lysosome. I'll just say to clean it up. Now you're ready for excretion. That's step 7. Lysosomal enzymes cleave T3 and T4 from thyroglobulin, and you just kind of like secrete it into the bloodstream. Cleave off. T3, T4, secrete. Okay. You know what I don't like is they don't really show you the whole thing ever. So I included a slide that shows you the T3, T4 on the thyroglobulin. So thyroglobulin is a globular shaped protein. You can see they use little well, let me draw the tyrosines the way they do. I'll just draw this here. DIT and DIT T4. There's one tyrosine. The iodine and iodine. And it's conjugated to another one. To another DIT. And that is what the lysosome does. It cleaves off just the T4, and that is what it's secreted. That's thyroid hormone. All right, so that's how it's synthesized, and I'm going to move on from thyroid to pancreas, unless there's questions on thyroid or parathyroid. Okay, well, yeah.
You said thyroid is like just the iodine? Is that what you said?
I said colloid could also be called thyroglobulin. Yeah. So right here, they say thyroglobulin colloid. Also, just back up a few slides. You see how they refer to colloid also as thyroglobulin?
Oh,
So on the quiz, I'm saying you could also call it thyroglobulin or colloid.
oh.
Okay?
So step number 7, you cleave off T3 and T4,
Yeah, you do.
and it goes into the blood.
That is correct. Alrighty. Pancreas. Where is it? Well, here's a picture of the abdominal cavity opened up. You get a full view of stomach, greater omentum. You can see the liver. But where's the pancreas? If you cut along the greater omentum, well, we haven't done stomach yet, but stomach, that's called the greater curvature. That's called the lesser curvature. So that mesentery that's attached to it is the lesser omentum, lesser curvature. So this is the greater curvature, and this big fat apron is the greater omentum. You cut along that edge, you're able to reflect back and see the stomach. And the space behind there is referred to as the lesser sac or omental bursa. So. So the pancreas is located behind the stomach, the space called the lesser sac or omental bursa. Basically what you see there. And you can't even see the full pancreas. That's the tail of the pancreas. This picture here, you have a more full view of the pancreas in its association with the duodenum. And so if you remove more structures, in this picture from the atlas, you can see the head of the pancreas looks kind of like an inverted tobacco pipe. And the head of the pancreas is in there, closely associated with the duodenum. And this is a very important part for digestion, but we're not concerned with that. So I just wanted to show you where the pancreas is. And let's look at the pancreatic islet. This is looking at the pancreas under a microscope, the pancreas actually has both exocrine and endocrine function. The exocrine function is for digestion. We'll get to it then. For now, the endocrine function I want you to be concerned with is how it regulates blood glucose through insulin and glucagon. Alpha and beta cells for the insulin and for the glucagon. But you have to locate those cells. You find them in those little pancreatic islets. That's why I want you to find it on that slide for lab today. So locate islet. You'll want to say islet, but you know what an island is, like islet. You know, the derivation of that word. Students still call it islet. Just know where the word came from. A little island of cells in a sea of other exocrine function. Okay, this little packet of cells is what we're looking for. Here's a picture I took. And it's always easy to identify if you can just find the right thing that sticks out. But there's some wrong things. That sticks out. That's not it. 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 This isn't it. I would say the best examples is this and that. Okay? You're looking for a clump of cells that stains a little bit different from everything else, and look for individual nuclei. Okay? And that's the nuclei of the clump of cells. And well, basically, what are the cell types in there? There's others besides alpha and beta, but you're not responsible for this one or that one. Just know that the beta cells of the islets will secrete the insulin, and the alpha cells will secrete the glucagon. So the physiology, the function of glucagon.
What were these other structures that you pointed out that you said that it's not them?
That looks like pancreatic duct. These look like blood vessel, pancreatic duct. So it's either pancreatic duct or blood vessels. That's a blood vessel. I can see red blood cells in there. Pancreatic duct will secrete all of the pancreatic enzymes that are useful for digestion right into the duodenum. Yeah, but the islets there is what we're talking about today. Okay, so just a list of things you should know for glucagon's function. Glucagon alpha cells versus insulin. Beta cells secreted. For glucagon, keyword, it's a hyperglycemic agent. It's going to increase blood glucose. So you're going to target the liver and skeletal muscle to do that. Those are the prime sources where you have glycogen, and you can kind of liberate it from the glycogen, and you can have glucose circulate. Targets: liver, skeletal muscle. I call it the exercise hormone because that's pretty much when you want to liberate the glucose from the glycogen and get it into the bloodstream to the working muscles during a workout. So activity level is important. So glycogenolysis, that's when you break down your glycogen, but this can also execute gluconeogenesis. And remember, I touched the difference between those two words. Let's write them down. Glycogenolysis. Endocytosis versus gluconeogenesis. So they both increase blood glucose, break down glycogen, break down fat and protein to get glucose. Okay. So cortisol is the main one that does it, but glucagon can as well. So you're going to increase the triglyceride breakdown, that's your fat stores. And so low blood glucose would stimulate glucagon's release. Increase triglyceride breakdown. You're basically breaking things down. So the word for that is catabolic. I'm going to put it in a word there. Now the other one, insulin, you can use the opposite word. This is your major anabolic hormone. Remember we talked about insulin-like growth factors stimulated to be released by growth hormone, and they were tissue building, and this is insulin. These aren't insulin-like growth factors. This is insulin, and what it does is it's a hypoglycemic agent. It lowers blood glucose. So this one and thyroid hormone are the only ones I know of that can lower blood glucose. Okay, so insulin decreases blood glucose. It targets skeletal muscle, adipose. Those are its targets. I want to add one, skeletal muscle adipose, also known liver. We talked about that before. The main one is skeletal muscle because your body, by mass, is mostly muscle. So if insulin's going to work, that tissue is the prime one. Your big muscle groups. All right. So insulin-sensitive tissues, those things there, the targets, they take up glucose from the blood into the cell. Okay. Cells, any of these, from any of these organs, so let's say that you have a cell, there's transporters that take glucose out of the blood into the cell, and they're actually called GLUT4. And researchers have shown that they kind of hang out inside the cell. These little vesicles that can transport glucose. But in the presence of high glucose, see after a large meal, if you act through insulin its receptor is on these target tissues. It will bind its receptor and trigger some molecular biology that causes the GLUT4 to translocate to the surface. And when it does so, it's able to take the glucose out of the blood into the cell for utilization. Okay. So, this is an insulin-dependent pathway. GLUT4 is the thing that actually brings it out of the blood in itself. So, once it gets in there, you utilize it for fuel. You can store it in glycogen or burn it, depending on what the body's needs are. You're increasing protein synthesis and triglyceride synthesis. Write that down. Also, right on the bottom, I know it's hard to see, protein and triglyceride, I'll just put TG for triglyceride synthesis. So let's say you're diabetic. Oh, I have that on the end. Let me leave that up there, Bill. But anyways, do you remember looking at this on the first day? So it was funny in summer. We get to the fourth day, and the first day seemed like an eternity ago. But we did look at this on Monday. And basically, it's insulin and glucagon again. Okay? Now, there's a study guide question for those of you who use it. I think the last question is this one, stating the effects of hormones have on fat, protein, glucose. And I thought I'd make life easy and just do it for you. And if you did it yourself, great. But here it is, hiding in my lecture. So go ahead and use that as a good review. I think what you'll notice is insulin is one of the few hormones that can actively lower it. But so can thyroid hormone because it boosts your metabolism. All the other ones increase it. So what does that tell you about the body? It needs glucose. Okay. I know there are a lot of low-carb diets out there. Maybe it helps you lose weight. I wouldn't try and work out. It's hard without glycogen. You need it. Okay? The body really needs glucose. You know another thing that can lower blood glucose, that's not a hormone? Exercise. Exercise does this, the GLUT4 translocation, without the insulin. Okay? You don't need insulin when you exercise because the exercise can lower it. So exercise, it really can help be used as a regimen to treat a diabetic patient. It should be used. And so diabetes, the one I was talking about is diabetes mellitus, which is a problem with insulin. Not to be confused with diabetes insipidus, which is a problem with ADH. So, that's a problem with ADH. No ADH. And let's think this through. Now you know what ADH does. Again, let's start there. Do you know what it does? I hope you do. Test this tomorrow. Water reabsorption, right? Okay, yes, water reabsorption. Okay, good. But okay, let's say now you don't have water reabsorption in this condition. What's going to happen to urine output? Is it going to increase or decrease if you can't reabsorb water from the kidney?
Increase.
It's going to increase. If you were to taste the urine, well, you know, I was told in a lecture that the ancient Egyptians used to taste the urine to test for diabetes. Okay, thankfully we don't have to do that. But you can see why it's referred to as tasteless urine. That's what this means. Okay. So diabetes mellitus is the problem with America. It's like, you know, 1 out of 3, 1 out of 4 people may contract it over their lifetime. It's a major epidemic. It's what I studied as a grad student. And, well, you can see why they call it sweet urine. The problem with insulin, you're insulin resistant. So what does that mean? Insulin is there, but it's not working. It's being secreted, not working. For whatever reason, and the reasons aren't clear, the insulin-sensitive tissues aren't sensitive to it. So it just doesn't do this as it's supposed to. You want insulin sensitivity to be increased, but if you become resistant to it, it's like it's there, it's knocking on the door, it just doesn't work. So you actually may hypersecrete insulin. You may have hyperinsulinemia. It's like there in boatloads trying to lower the glucose, but it can't. It's there, but it's not working. And this is called the type 2 diabetes, which is the much more prevalent kind, where insulin is there, but is not working. And it may be preceded by a period of hyperinsulinemia, kind of a pre-diabetes phase. Hyperinsulinemia. That means a lot of insulin. It's there, it doesn't work. And I mean, this is a chance whenever this, if you're doing preventative things like getting tested for it, the blood draw, fasting glucose, or hemoglobin A1C, I think that's the one they look at the most. It's kind of your average blood glucose levels for a month. They can tell based on that. But anyways, the physiology is it's there and it doesn't work. There is type 1, which usually presents in juveniles. This one you usually see in middle-aged adults. Okay. So type 1, they used to call this juvenile diabetes because that's back in the 80s they observed it in young people. I'm sorry, type 1. It used to be called juvenile diabetes. Today we just call it type 1. This one used to be called adult onset diabetes. I didn't get that right. I pointed to the wrong thing when I said it. Adult onset. This one, usually if you contract a virus, and the virus mimics beta cell proteins, well, the antibody you make against it attacks that virus, but also the beta cells, and you destroy insulin function. So you don't have insulin. So type 1 is the pancreas stops secreting insulin. It's not there. So you're dependent upon injections. And they notice this in a small group of juveniles. Well, the scary thing is the type 2, we're starting to see it in younger people. It usually takes decades of your life before you develop an insulin resistance. And now, due to, I guess, poor lifestyle, decreased activity, poor diet, this usually is a problem. Usually fat consumption is a big problem that causes diabetes. People usually don't think about that. Okay. My research diet is a very low-fat diet that really helps people. But think about this. Okay, so if you have diabetes, this doesn't happen. Okay. So the great thing is, if insulin doesn't work well and you're trying to treat a diabetic patient, exercise, again, can do this without the need for insulin. So, and it also increases insulin sensitivity. So not only does it help control the glucose levels, exercise makes insulin work better in a type 2 diabetic patient. So it should be considered as part of the treating regimen for any diabetic patient. Okay. That was it. The test is tomorrow. What I'm going to do now, we'll do a break. When we come back from break,